The purpose of this discussion is to give you an overview of some of the more complex elements of population genetics. And again, it is just an overview, but I wanted to start with the more simple version of the Hardy Weinberg equation, which is kind of like the beginner's insight into population genetics. And to do that, you should already be very familiar with it. You should be comfortable doing equations, and if you are given genotype counts, you should be able to turn those into allele frequencies. Once you have those allele frequencies, you should be able to plug those into the Hardy-Weinberg equation and then determine whether or not there is an evolutionary force acting on that gene. And then perhaps even you should be able to extend beyond that and say, okay, if it was a natural selection force, what of the three types of natural selection are we looking at? Is it a directional selection force? Is it a balancing selection force, which is also known as overdominance? Or is it a diversifying selection force, which in the two allele version is called underdominance? Okay, so hopefully you're somewhat familiar with those. Now, I wanted to get just a little bit into how the equation was derived. We talked about it in general terms, but I'm going to be more specific here. And there's going to be a little bit of mathematical notation. I'm not really interested in you memorizing or mastering the mathematical notation. This is for the math of probabilities. But I do want you to understand the concept, because if you understand the concept, then you can extend it beyond this specific example, which is the most common one, right? Where we're looking at a diploid organism with two alleles, okay? Now, a couple of critical things. First, when we do this, we are assuming independence. That's the assumption, meaning that whether you get a dominant allele does not have an impact on what your second allele is. So that dominant allele does not bring in the recessive allele or vice versa. They're not associated. And in sexually reproducing organisms, because of random assortment and because of two different parents, then that's a pretty valid assumption. Now, if we have selfing, then independence may not be a completely valid assumption. If there's not random assortment, then we need to look at if we're looking at two sites, two different genes. Okay, but for the most part, that's a very valid assumption. So it really is like drawing two marbles from a jar. What you get the first time does not have any impact on what you get the second time. Now, you might be thinking there is a problem or a potential violation of that assumption. If we have a finite number of balls in here, and it becomes readily apparent if there's a very, very small number. So for instance, even if we had an even number of balls, if there were only like, say, 8 balls, 4 red ones and 4 blue ones, then when I withdrew the first ball, let's say I get a red one, that's going to have an impact, and probably in this case a significant impact, on what the possible outcomes are for the second draw, right? So if we're looking at that as two random draws to get random alleles. So realize this assumption of independence requires maybe not an infinite population, but at least a somewhat large one, so that by removing one allele, by creating the first gamete, we're not biasing the pool of alleles that is left. But again, for reasonably sized populations, that's a very valid assumption. And then the last thing, it's probably worth actually putting it in here, so let me do that. We are also assuming that, and we're going to represent our frequencies. When we're calculating probabilities, our frequencies are going to be numbers between 0, representing a 0% chance, and 1. And so anything like 0.2 is 20%, 0.5 is 50%. The math works when you have them representative frequencies between 0 and 1. Okay, again, that's hopefully you're somewhat comfortable with that already. Now let's look at the details. When we are looking at two things occurring and we need both of them to fulfill the expectation, that is called the union, and again, we're assuming independence, so the union of two independent events. So if I was drawing from marbles, it is like saying, what are the chances that if I draw 2 marbles from the jar, I get a red one, and then the second draw is also a red one. And that's just like getting 2 alleles together. What's the probability of getting a big A allele and another big A allele in the same individual? Okay. And so the union is expressed like this, the probability of A and, right, so both of those events occurring sequentially. What's the probability of getting an A and then in the second draw getting a B? Well, that would be the probability of A union of B, right, which is simply the product of those probabilities. So if we're at 50-50, then the probability of getting an A and then a B in a single event would be 0.5 times 0.5 or 25%, right? And you can use this, you can actually extend this to many things beyond biology. So if you understand probability, it's pretty easy. So you could even say, what are the probabilities of rolling a snake eyes, right? A 1 and a 1 on 2 die. And you could say, well, the probability of getting a 1 is 1 in 6. The probability of getting a 1 on the second die is also 1 in 6. So it'd be 1 in 6 times 1 in 6, or 1 out of every 36 rolls on average, you would expect to get snake eyes. So this extends beyond this, and it's just a simple probability. And so Hardy and Weinberg's major insight was recognizing that we could treat alleles in a population, given certain conditions, which are those 5 conditions you learned in the last one. But given those conditions, we can treat alleles as random independent events. And so figuring out where they end up in offspring is simply the outcome of these simple laws of probability. Okay, so the product of those probabilities represents the union of those 2 events. Now, the second important probability calculation for us is what we call the intersection. And the intersection is when we get an A or a B. So what is the probability that I get an A or a B? That's represented by this little upside down U symbol. That is the intersection of events. And to figure that out, we do the probability of A and the probability of B. So very simply, and we'll look at how this is applied when we look at the Hardy-Weinberg equation here in just a bit. But very simply, you say, if I draw a marble, what is the probability that I get a red or I get a blue? And if those are your only 2 options, then that's very simple, right? The probability of A, which is 50%, plus the probability of B is 50%. So you have a 100% chance of getting either a red or a blue, okay? So this is the intersection of 2 sequential events, okay? Now, so we already defined the frequency of A as this lowercase p, and it could have been any number, but that's, or any letter to stand in for the frequency of A, but that's what was used. So the probability of getting 2 big A alleles together is simply the union, right, of those events. So that's where p squared comes from. Okay, hopefully that makes sense in your head. And Q is the same, right? It's easy. The frequency of A, then the probability of getting 2 A's together is simply the union of whatever the frequency of those little a's are. So that's where the P and the Q come from. Now, the 2PQ is a little trickier, but not much, because there are 2 ways we can get a heterozygote. So if we want to figure out heterozygote, it doesn't matter if dad donates the big A or dad donates little a, or vice versa, right? Mom little a, mom big. It doesn't matter. So there are 2 ways we can make a heterozygote. And so this is one of those or statements here, right? So to figure out the heterozygote frequency, we need the probability of making a heterozygote this way added to whatever the probability is of making a heterozygote the other way. And again, in a 2 allele, autosomal diploid 2 allele system, those numbers, p squared, q squared, 2pq, are always going to sum to 1. Now, I went through that derivation. If you remember the equation and memorize the equation, or able to put in your notes and look it up for the exam, that's fine. That's part of the way, at least. But I hope you understand the concept, because you might see something on an exam like this. So let's modify the system. Let's change it from a 2 allele example to a 3 allele example. Now, the organisms are still diploid, so any one organism can only carry 2 alleles, but there are 3 possible alleles out there in the population. And so if we want to look at the frequency, we could look at something, and a nice convenient example of this is ABO blood typing system. Now, if you recognize ABO, you'll know, maybe you know some of the details about it. But basically, we're looking for the presence or absence of certain proteins on the surface of red blood cells, and that's where the ABO blood typing comes from. And it's important for blood donation and compatibility when we're looking at donors versus recipients of blood. So it has some real-life applications. But is there any natural selection acting on the ABO blood type system? Well, first, to do that, we would need to be able to calculate where it should be if there is no natural selection acting on it. So let's see if you can derive that. So I'm going to give you a little bit of backup, or a little bit of background, and then I'm going to ask you to pause the video and see if you can figure it out. Okay? So, with 3 alleles, there are more diploid combinations that we can make, right? If you have 2 alleles, there are basically only 3 diploid possibilities. You can be homozygous for one allele, homozygous for the other allele, or you can be heterozygous. And that's the extent of all of the possibilities. But when we have more than 2 alleles, there are suddenly more than 3 possible outcomes. And here's an equation. You don't need to memorize it. It's handy if you want to look at it, but you can always refer to it, right? N is the number of alleles, then we can exactly calculate the number of possible genotypes. So with 3 alleles, this becomes a 3 plus 1, or 4 times 3, 12, divided by 2. So there are 6 possible alleles. And you can figure this out pretty simply by just looking at all the combinations, right? So you can be homozygous for the A blood type. You can be homozygous for the B blood type, the O blood type. Those are all your homozygotes. And then there are 3 possible heterozygotes represented here. Now, you're thinking, well, there aren't that many blood types, because the way we classify blood type, a big A and a big A-O is the same blood type, because they are both sensitive to that A antigen and will reject the B antigen. But O is a universal donor. It doesn't conflict with either A or B. So it's basically, essentially, A and B are codominant. Both of the phenotypes are expressed if you're AB, and O is recessive to both of those. Now, here's where I want you to pause it. So if P equals the frequency of the A allele, Q equals the frequency of the B allele, and R equals the frequency of the O allele, how could you calculate out a prediction, a modification of the Hardy Weinberg equation to represent all of the individuals in the population? And remember, with Hardy Weinberg, it's a prediction for if there is no selection. So you need to figure out what the frequency of this homozygote would be. Hopefully that's pretty straightforward from what we've already done. The frequency of this homozygote and the frequency of that homozygote, add them all together, and then figure out the frequencies of the heterozygote. So pause the video now and see if you can figure it out. Okay, so hopefully you're back now. I just paused for a second, but if you're not, pause the video and really see if you can do it. So I'm going to put it in here. Oops, let's modify this. So we'll just put it in the middle. I maybe have to make my font a little bit smaller. So it's kind of easy, right? The first part's very similar, right? So it would still be P squared. I'll make those superscript in a little bit. Q squared plus R squared, which we're just using R, and again, you could have picked any letter, but since R comes after P and Q, it works well, right? So this would be the equation for figuring out the contributions of all of the homozygotes. But we still need to figure out the heterozygotes, okay? So, and if you look at it, it's the same way. A and B is the same as we were calculating before, so it would be 2 times the probability of A, which we've told you is a P, and the probability of B, which is a Q. Okay, let's make this much smaller so it will fit on here. And then we just extend that. So that's the probability of, sorry if I don't talk long enough, it thinks that something's wrong. That's the probability of the O allele getting together with the B allele. So that would be the probability of BO. And then finally, we would add in the probability of the last one. So that'd be 2 RP. And this is always going to equal 1 if we've done everything correctly. But if we were given estimates of all of these genotypes from the population, we could plug them into the equation and see if they match the original estimates, right? If everything here matched the original one, then there'd be no natural selection. So we could test for natural selection. Now recognize that the data we usually get for blood types would probably not be sufficient for that test, because you'd get results back that's saying, oh, you're A blood type. You wouldn't know whether those individuals, how many of them were big A, and how many of them were big A, O. So you would need to do more of a genotyping rather than just a blood typing test to make sure that you were able to capture all of the heterozygotes. Because of the dominance and recessive system here, some of them don't recognize, or some of them manifest as the same phenotype. Okay, so just be aware, understand these basic probabilities, how they work, and be able to extend them maybe with some simpler examples. So maybe think about, what if you had a triploid population? What if you had only 2 alleles still, but maybe there was a triploid population, so they had, I don't know, and triploid populations are a little weird because they don't really have three parents. They more often are making copies of themselves. So triploid may be a little bit of a not practical example because you don't have three parents making one offspring. But what if we had a tetraploid where you could have four different alleles, right? So just start to think about those, and maybe with some simple extensions, you could figure out Hardy Weinberg equations and how it could apply to other things. But still, Hardy Weinberg is looking at one single locus. No matter how many alleles we have, we really only do one locus when we're one site, right? One gene when we're doing Hardy Weinberg equilibrium. And so people wanted to be able to extend this to more than one gene, more than one locus. And so what they realized is that they could maybe come up with kind of a big picture overview of genetic diversity. And the most common form of this is what we call heterozygosity. Now, for our simple examples that we've looked at, heterozygosity is simply equal to 2pq, or the frequency of heterozygotes, right? For one allele with one population we're looking at, that's the heterozygosity for that gene. But if we have multiple alleles, it gets a little bit more complicated, right? So if we wanted to figure out the heterozygosity for the blood typing system, we would need to count up all the heterozygotes and say what frequency of the populations are AB, what frequency are AO, and what frequency are BO. And those all added together would represent the frequency of the heterozygotes. And so this in the paper that you're supposed to read for today, that's how they represent this. It's 2 times the frequency of one heterozygote times the frequency of the other heterozygote, but then we need to do all combinations of alleles here. Or an alternative to that is used to do 1 minus the sum of all of the homozygotes, right? So this would be the A blood type plus the B blood type plus the O blood type when they're homozygous for those, and subtract all those from 1, and that will give you the heterozygosity. So sometimes it's easier to calculate in different ways. But basically, it's just how many, it's a way to figure out what frequency of the population is heterozygous for any one allele. Sorry, for any one gene, right? It doesn't really make sense to be heterozygous for an allele. Sorry, I misspoke. Heterozygous for any one gene. And that is your heterozygosity. And so it's an estimate of genetic diversity for that gene, right? Okay, but it's only for that one gene. So if we want to extend that beyond that one gene, this is where we get now the average heterozygosity, which is represented by this bar over the big H, which is heterozygosity for one site. And so basically, we would do a count through gathering data, through genotyping. There are multiple ways we could do this. But we gather lots of data, and we look at, maybe we've even picked genes randomly. Let's pick 50 genes randomly and see what the levels of heterozygosity are for each of those 50 genes, and then give us an average. And that average represents genetic diversity. The higher the heterozygosity is, the more genetic diversity there is. And so signs of healthy populations have relatively high heterozygosity. One of the main things that can decrease the heterozygosity of a population is some sort of drastic drop in population size, because then you have genetic drift that randomly removes diversity, and it does it across the genome, not just for one gene. And so lower than expected heterozygosity across a genome usually represents some sort of extinction pressure, sickness pressure, some sort of population pressure where we've got a much lower, or at least recently, there was a much lower population than is good for that organism. Now, the paper that you read for today also goes into a lot more detail. We're not going to get into all those, but realize that there are exceptions to this rule. So for autosomal genes, it's a very straight calculation no matter what sex the individual is, whether they are male XY or female XX. It doesn't matter for the autosomal genes. But for the X and Y genes, right, these sex-linked chromosomes, the sex chromosomes, it makes a difference because basically the Y is a placeholder. There's really no genetic material, at least very little on it. And so the frequency for males is different than the frequency for females. I'm not going to go into the details. It's not really critical for this class that you're able to calculate or know any of that, but just be aware that the sex-linked genes are a violation of Hardy Weinberg in individuals like humans that have chromosomal sex determination. Now, that's not all organisms, but many organisms, including mammals, many other organisms, but that have some sort, or either just like an XY system or a close variant thereof. Okay, so then we might need to consider genetics for, so if something is found on the X chromosome, it might have slightly different considerations than all the other genes that are on the autosomal chromosomes. All right, now we're going to look at some of the values that are used in population genetics, and we're just going to give you a bird's eye view. This is a little bit more advanced, but we're not going to get into the derivation, the details. You just need to know the basic definition and what they are used for. One of the very common, there are three common values that are often estimated in population genetics. One is theta, okay? And theta is an estimate of genetic diversity. If everything is neutral, then theta, the genetic diversity, should, for diploid populations, should be 4 times the population size, everyone that reproduces, so we may even want to put a little e under here for effective population size, which is only the individuals that contribute to the next generation. But it would be 4 times the population size times the mutation rate. That should be what the genetic diversity is. And so if we are seeing a genetic diversity that is not lining up with this, then that could be a sign of some sort of selection force for that organism. Okay? So mutations are usually based on a per base pair per generation, but there are slightly different calculations and different ways to do that. But just know what the N stands for, population size. This Greek letter mu stands for mutation rate. Convenient, right? It makes sense. Now, our largest, biggest sources of data today are single nucleotide polymorphisms, or SNPs. A SNP is simply a location in the chromosome where there's variation amongst individuals. And it might be only a small, tiny fraction of the population. Maybe only 0.03% of the population has an A at that position and everybody has a G or a T, or everyone has Gs. But it's still a polymorphism, a variant there. And oftentimes they're at higher levels. And these are the most common type of mutations. They're caused by a single mistake during DNA replication. And so they're most widely relied upon. And when we look at all of the SNPs that have been sampled and identified across the human population, it turns out that approximately 85% of them are randomly, evenly distributed without respect to geography. So there's really no rhyme or reason. There's no so these are probably SNPs that arose fairly anciently and have just been kind of moved and randomly evolving, not good or bad. There's no natural selection force on them. And so they are just in the population generally and kind of spread evenly across all subpopulations. However, the remaining 15% show some sort of a demographic signal, meaning that they are different in different areas and maybe even unique to specific areas. And in fact, for people that are doing the DNA testing where you can send a sample of your DNA in and you get results back saying, oh, you're 5% Chinese and 4% African and 3% Eastern European or whatever, they have identified SNPs by looking at indigenous populations. And that's hard for some areas of the world because some areas have seen so much migration and movement in and out that it's really hard to identify people that have been in that area for a very, very long time and have only ancestors from that area. But as much as they can do it, and there are ways to get around it even if you don't have indigenous populations, but you need large data sets. And so the DNA tests rely on identifying these SNPs that are unique and specific to certain areas. But there are other sources of data also that can be used, and these can be valuable, particularly in very specific types of populations. So we can have copy number variants that result from gene duplications, and maybe even a large number of copies that are vary. We can have microsatellites, which are often short tandem repeats or triad repeats, but short repeats that really evolve quickly because it's easy to make mistakes when you're trying to copy TC, 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 TC over and over again, or any other two tandem repeats. So microsatellites are amongst the fastest evolving molecular markers, but all of these are called molecular markers. And in many genomes, especially studied organisms, modeled organisms, humans, mice, Arabidopsis, yeast, right? Arabidopsis is a plant, if you're not familiar with that, a model plant. We know their genomes very, very well and even have many, many genomes. So we've identified many SNPs, many microsatellites, and other copy number variants across those genomes. And so we can use them as markers or identifiers of gene location or loci. And so we might have many, many different unique things, and I've only done, a dozen or two here across a chromosome, but there might be several hundred that we recognize as polymorphisms and useful sources of data for that chromosome across an entire population. All right, two more statistics that you need to know. The first is called the D statistic, or also Tajima's D statistic, because this was the guy that first proposed it, a Japanese researcher. And a D statistic is simply a way to get two independent estimates of genetic diversity. Remember, theta is an estimate of genetic diversity, and there are different ways to calculate it. And so if we use one calculation, don't worry about the subscripts here, they're just to denote the different ways to calculate genetic diversity. We calculate genetic diversity, and then we do statistical tests. If they are significantly different from one another, then suddenly there's some force, oftentimes natural selection, in fact, mostly natural selection, that's been acting on that locus or that site in the chromosome. So the D statistic then becomes a way to estimate whether or not there's been natural selection working on a certain site. The last value that you need to be familiar with, and again, there's a lot more, we can go into many, many more details about these, but for our class, as long as you know what they're used for, that's all you need to know. Okay, so for the F statistic, this becomes a measure of population structure, and there are actually multiple ones. Some of them use individuals versus the whole population, or individuals versus subpopulations. The one that is most frequently used, and the only one that we are going to define and talk about, is called FST. That's a subscript. So FST is a way where we estimate the heterozygosity, maybe take many genetic samples and estimate what the heterozygosity is in one certain subpopulation. And then we compare it to the overall genetic diversity in the entire population. So this is a diagram map of different regions of polar bear range. We have individuals that live across Hudson Bay, individuals that live up in Norway. I guess this is just one sub. This looks like only the North American ones, right? So across Greenland, Arctic Circle. But anyway, we could measure heterozygosity in this population, compare it to the overall population. Measure it in this subpopulation, compare it to the overall population. And if there's a discrepancy, then that becomes a sign for, again, some sort of selection evolutionary pressure that is acting on that subpopulation. And there's statistic involved, whether they are, significantly different or just different based on what we would expect by chance. We have to do some statistical estimates to determine whether it is significant or not. But this then becomes a very powerful tool for determining whether there are different selection pressures on an individual gene based on where the organisms are living. So it's a really useful number to calculate for populations where we have some sort of geographical structure, and then we want to determine whether that geographical structure also correlates with genetic diversity.